hit the record button. Yes, so I would like us to welcome Dr. Andy Booth. I will introduce him shortly. Andy Booth is the chief scientist at the Sintef Ocean Department of Climate and Environment. Dr. Booth is an environmental chemist with significant experience in developing analytical approaches and studying the fate and effects of contaminants such as micro and nanoplastic, nanomaterials, persistent organic pollutants, POPs, crude oils, and other emerging pollutants. Recently, he has focused on investigating the degradation and leaching of plastic-associated chemicals. He collaborates closely with a wide range of researchers from other disciplines, including toxicology, microbiology, materials science, and social sciences. He enjoys contributing to the development of new technologies and innovations, especially through the safer by design approaches. He is an active member of the national and internal research community on plastics, being a member of the scientific board of the Micro Conference Series, an expert member for the microplastic in the EU Marine Strategy Framework Directive Technical Group on Marine Litter, and co-chair in 2017 to 2021. Of the International Council for Exploration of the Seas Working Group on Marine Litter. He has acted as a project proposal reviewer for the national funding agencies in Sweden, Germany, France, Belgium, and Hong Kong. He has coordinated or been a partner in over 15 projects focused on issues related to plastic pollution in the environment, with financial support from the Norwegian Research Council, the EU, JPI Oceans, and FRAM Centret. He has an H index of 34 and has published over 80 peer-reviewed articles, more than 30 of them on plastic-related topics, and 6 book chapters, all on plastic-related topics, with over 3,500 citations. And he also regularly publishes in national and international popular science channels, and he contributes to exhibitions. Today, Dr. Booth will tell us just how dangerous is nano and microplastics. So, Dr. Booth, the floor is yours.
Thanks so much. I wasn't expecting you to read all of the bio information. Hi, Sam. Nice to see you.
Hi.
So thank you for that very long.
Apologies, but yeah, we wanted people to get to know you.
Yeah.
Cool. This I have a presentation that will take I don't know, around 45 minutes, hopefully. I might not be able to, I'll keep an eye on the time, but and then yeah, hopefully this will just give you a bit of a sort of a flavor of some of the work that we've been doing in the last few years. Sam, I'm sorry there isn't any Go Jelly stuff in here, but maybe we can save that one and you can be a guest another time. So I share my screen. Presentation. Great. Can you see my screen okay?
Yeah? Yes.
Excellent. Okay, so this is a presentation that I gave earlier this year in June. I was an invited keynote speaker at a conference in Norway. And I was talking to toxicologists. It was the Norwegian Environmental Toxicology Conference. My background is chemistry. So, well, you'll see. I'm an environmental analytical chemist. Like you mentioned, I've worked quite extensively with micro and nanoplastic from an environmental fate and effects perspective for like 10 years. But I stopped studying biology when I was 16. This is me when I was 16. I had hair and an interesting choice in t-shirts. But I've worked closely with toxicologists throughout my research career, and I've done basic toxicology myself. So what I sort of hope this presentation will do is provide a bit of a current status on the research, on the risks associated with micro and nanoplastic, and specifically try to highlight how we can improve the risk assessment process for these pollutants going forward. So when you look in the media, quite often you will see these sorts of headlines, you know, with the words toxic, poisoning, harmful, when discussing or having articles about plastic and microplastic. But is this anchored really in the science right now? What we do know is that there's a lot of this micro nanoplastic around, but is it really that harmful? And is this increasing societal acceptance that micro nanoplastic represents an environmental and a human health risk? Is it based on scientific fact? That's what we're going to sort of explore a little bit today. So we're going to go right back to the beginning, because I don't quite know everyone's background here. So I've tried to keep this as sort of reasonably accessible, I hope, because it's aquatic pollutants. So I'm guessing most people have heard of microplastic. But if you think, what is plastic? Most of us would think of the plastic goods that we have at home, food packaging, plastic bags, that type of thing. And when we think about plastic in the environment, we, you know, might draw to mind images like you have on the left here. And if we really pushed on it, we might start to go, well, we've heard of PET, polyethylene terephthalate, plastic bottles, and polyethylene. You know, we think of the polymer type, and that's about as detailed as I think most people tend to go. But the reality is that pretty much every consumer product that is made of plastic is comprised of a polymer plus some combination and some concentration of additive chemicals. These are added to plastic to impart particular properties. You know, it might be flexibility, it might be to protect the plastic, it might be to give it a particular color. But all of these things are chemicals that we're adding. And the sort of broader thing, this plasticizers, these antioxidants, UV stabilizers, flame retardants, and colorants, as I said. And the important thing to consider here is that the bulk of these additives are not covalently bonded to the polymer, so they're sort of dispersed throughout the polymer matrix, and that gives them potential for mobility. There's all sorts of numbers out in the literature about how many different chemicals are used as additives. I picked this one. I can't remember exactly which study it was from, but you know, we're talking thousands of different chemicals at least. And these are just some examples. And I picked these ones out partly because you might have heard of some of them, like bisphenol A, perhaps triclosan, and then we've got examples of things like styrene here, which is the building block and a residual chemical from the production of polystyrene. But these are all chemicals, as you can see, that vary massively in their size. The additional atoms that are included in the molecular structure. So it's a big diversity. That's the point that I'm trying to make here, both in terms of sort of size and chemical properties. So we'll come back to additives and chemicals associated with plastic a little bit later on. But the first place that I thought we would start would be, where can we be confident about microplastic? What is it that we feel that we know well about it? And in my opinion, I think we're actually getting to the stage now that we have pretty good methods and instrumentation that we can apply for determining environmental levels of microplastic, at least for particles bigger than 10 micron. And I pulled out this paper from one of Melanie Bergman's, this picture from one of Melanie Bergman's recent papers. And it gives a nice overview of all the different types of matrices. This is more focused on the marine environment, but we're getting to the same stage with terrestrial systems. We're starting to have lots of studies, and we're able to start to get concentration ranges for different environmental compartments in terms of microplastic. I mean, you can see from the numbers, these range massively, but that also reflects on multiple things, different methodologies, different quality of the research, just generally variation in levels, because these are based on data from all over the globe, northern hemisphere, southern hemisphere, tropics to poles, sea floors to the tops of mountains, effectively. Of course, there's going to be variation. But we also know that, the vast majority of microplastic particles that we find in the environment are partially degraded in some way. And by this, they fall under the term secondary microplastic, which is plastics which are, or plastic particles, which are derived, their fragmentation or degradation products from larger plastic items, as opposed to primary microplastic particles, which things like the pellets and microbeads that are added to certain consumer products. Those are micron-sized plastic particles that are specifically manufactured in that size and used for a particular purpose. But most of what we see is these partially degraded particles, and we're starting to get a good overview of the mechanisms that are driving the degradation process. The primary one is actually UV degradation, because this really changes the properties of the material. When you combine UV with something like physical or mechanical degradation, that could be like wave action, it can be all sorts of different things. Then you start to get this fragmentation process. You hear a lot about like biodegradation and microbial degradation, but for all of the conventional thermoplastics, the understanding is that no microbial degradation is going to proceed until you get down to the molecular level. So you've got to have a lot of other degradation mechanisms occurring first. There are a few other oxidation pathways for plastics. Hydrolysis is technically relevant for certain polymer types, for example, polyesters, but under environmental conditions, that's very rare. And of course, heat could potentially be important, but again, it's rare that there's anywhere globally that the heat is high enough to really be, to drive degradation. The thing probably to keep in mind with this degradation is that it's an irreversible process, and it actually can cause quite major changes in material properties. So that includes things like molecular mass, the structure, the mechanical strength. Also, I haven't listed it here, but also chemical changes, these oxidation processes, they're changing the chemistry, particularly on the surface, that's in contact with the environment. And it's this that leads to this fragmentation process where you go from a macroplastic litter item through the microplastic size range, nanoplastic, and down eventually to polymer fragments. And so, I've highlighted degradation. Is it that important? We're starting to think that it certainly would play a role. So we had a study that came out last year, I think it was, last year, where we were looking at textile fibers. And this was UV degradation studies. And we had polyamide or nylon, that's the material on the left. Then we had polyester, it's the second material. We had some wool as a control, and we had polyacrylic as the third material. We had different colors of some of these as well. And we saw some quite significant changes for some of the particles. And without going into detail, we get on the top right, you can see that UV exposure changes the PET, the polyester, from a white to a sort of a brown color. So we're getting, it's evidence of oxidation. And if we look back at the PET under the scanning electron microscopy images, you can see that, we go from the top of the column over 14 days down to 10 months. And what we're seeing is, quite strong changes in the surface properties, and they become very, very brittle as well. So we get a lot of fragmentation. We saw the same for the polyamide. It was even more sort of surface changes. We're getting really big changes in the morphology of the surface. And if we're starting to think about toxicology, it's the surface of particles that organisms are interacting with. These are quite fundamental changes. Of course, it's not the same for all polymer types necessarily. So the polyacrylic, which is the bottom right, you can see after 10 months, there was virtually no change, just maybe the start of some evidence of degradation. So what we know about environmental microplastic is that it's irregular shaped, it's partially degraded. It represents a continuum of sizes, shapes, and densities. It comprises a wide range of polymer types. And across all of the plastic litter of all sizes, there's a vast range of plastic associated chemicals that are, part of that pollution. So this brings us to the sort of microplastic paradox that we need to start to think about when we're doing toxicological studies or trying to assess the hazard associated with microplastic. And because of this fragmentation, there was a nice study by Bart Kuhlman's group in the Netherlands a couple of years ago. They basically sort of demonstrated that no two microplastic particles would ever be identical. You need two populations that would each contain 10 to the 5 particles to get an average set of properties that would be the same. So that's the kind of complexity we're dealing with this particular form of pollutant, and it's quite a long way away from individual chemicals or metals or as pollutants. It really represents a sort of complex mixture of particulate and chemical pollutant. And it's this that poses a problem when it comes to hazard assessment. What microplastics should, test materials should we use in toxicity tests? And I think this is reflected, this is a little bit out of date now. I should update this slide, but at least until sort of 2019, 2020, it's very clear that hazard assessments, so toxicological studies, accumulation studies, they've really not kept pace with the number of studies focused on measuring microplastic levels in the environment, so what I would call the exposure assessment. So, by this point, there were hundreds and hundreds of papers on microplastic exposure assessments, but there was a 2019 study that identified only 46 publications reporting microplastic toxicity data. In the same year, there was a Ribeiro et al. Paper that was looking at accumulation studies, and you can see there that there's a few more, but it's not huge amounts more. So we don't actually, and this, it's changing. There have been more studies coming in the last 2 or 3 years. It's a fast-moving field, but if we think back to those headlines in the newspapers that microplastic is toxic, and we've got 46 studies claiming or demonstrating there may or may not be some toxicity, then and each of these studies has not used environmental plastic, it starts to become a bit of a question about whether we can make these statements. So when we look a little bit at these, the studies that are available on toxicity, there are reports or evidence of sublethal effects. Most of the studies are focused on invertebrate species. There were a few fish studies. It's grown again in the last few years. And there's a range of sublethal effects have been reported. I've just listed a few of them here, but you know, some of them are, you know, quite important. You know, you've got developmental abnormalities, you've got reproductive abnormalities, things like that. And of course, you have, you know, things like reduced feeding and weight loss that, you know, have an impact on how successful an organism is in its natural environment. But overall, we still have very limited studies when it comes to toxicology. When we think back to the complexity of microplastic as a pollutant, there are a large number of potential drivers for the observed toxicological responses. That includes polymer type, the size of the particle, the shape, surface characteristics. I think there's been 1 or 2 studies on the crystallinity of the polymer. There's all of these additives that are and other chemicals associated with plastic that could be a potential driver, and then surface chemistry and surface characteristics, shall we say. So the particles and contaminants on the surface, any absorbed molecules, changes in the surface chemistry due to degradation, and potentially the presence of microorganisms that colonize the surface. So the eagle-eyed amongst you might have noticed that I think the title of the presentation included the word nanoplastics, and I haven't really talked about these so much. And these are a bit of the sort of elephant in the room, because we kind of talk a lot about them, but we know actually very little about them. And I'll sort of explain why that is now. So we're increasingly seeing from the exposure assessment studies, when people have gone out into the environment, taken their water, sediments, biota samples, that typically as you go down in size, the number of particles, the relative abundance of particles increases almost exponentially as you go from sort of, you know, 1 millimeter down to 10 micron. And from this, we would then sort of infer that as you go below 10 micron in size and into the nanoscale, that, you know, this sort of exponential increase would continue. The reality is there's potential for very high numbers of nanoplastics in the environment, but we lack what's the best way to put this? The extraction techniques, the sample processing methodologies, and the analysis techniques to be able to look at the nanofraction properly. There's been a few studies, as we'll see in a second. It's kind of interesting because we think that nanoscale particles are likely to be a greater risk than micron scale particles. There's a lot of work that's been done on nanomaterials in the last 20 years from an environmental and human health perspective. We know that there are risks associated with particles at that scale. And the reason why microplastic is thought to be maybe less of a risk is that those particles are, in theory, too large to cross most biological barriers, i.e. if you ingest it and it's going through the body, the gut is not going to allow large micron-sized particles to pass through and lead to accumulation. So, but we do know that the nanoscale particles can pass through biological barriers. It's a bit of a grey area right now in terms of exactly where this sort of cutoff is and what size can particles transfer and accumulate into the body. And that's one focus of research at the moment. But now we're talking about nanoparticles or nanoscale particles and microplastic particles. And I find that it's really hard to get your head around the sizes involved here. And this will help sort of explain a little bit why we aren't anywhere near as advanced with nanoplastic research as we are with microplastic. So I've put gold nanoparticles here on the left and some microplastic on the right. And at a quick glance, they don't look too dissimilar. But I think the problem that many of us have is the scaling issue. Is that microplastic is nothing like nanoscale particles. And one way to try and sort of visualize this is that a 200 micrometer particle, so a 200 micron microplastic particle, which is nowhere near the upper range of what is considered a microplastic particle, that's 5 millimeters. That's 10,000 times larger than a 20 nanometer particle. So when you try and scale that, that's the equivalent of a sand grain compared to a blue whale. So we have good techniques for analyzing the blue whale and getting it out of environmental matrices. We're okay there, but we are not so good at doing it with sand yet. And I think that's something that a lot of folks struggle to sort of comprehend, just how challenging it is, and that we can't use the same methodologies, we can't use the same analysis techniques, because they just don't cover this huge size range. Excuse me. So that leads us to the issue of this sort of methodology gap for submicron and nanoparticles. We struggle to go much below 1 micron at the moment in any sort of reliable, robust way. And so there's quite a lot of work at the moment focused on, including in our own labs, focused on trying to develop reference materials and test materials so that we can develop the methods we need in that for that size range here. So the nanoplastic goal, as we call it, we want to be able to take an environmental sample, that could be water, soil, sediments, whatever it is. We're going to need some form of extraction and digestion step to remove the background matrix. We need to be able to concentrate this. We might want to separate, because we're probably, well, you would need to separate because there will be larger particles there as well. If we want a nano fraction, we've got to separate out the nano fraction. And then you've got to have the analysis technique. So we need to be able to image, we need to be able to do size analysis, we need to be able to do identification, and all of that is what leads us towards some form of quantification. As of 2021, there are only 5 studies that have attempted to report nanoparticle exposure, nanoplastic exposure levels. I think there's only maybe 1 or 2 more since then that I've seen. And I'm sure the authors wouldn't hate me for saying that, you know, these are the first studies on nanoplastic, and there are some limitations with the approaches. You know, these will get better, but, you know, so we don't have a lot of information when it comes to nanoplastic. So when we come back to toxicity testing of these types of particles, one of the key issues is a lack of environmentally relevant reference materials or test materials that sort of replicate more closely the micro and potentially nanoplastic particles we will find in the environment. There's also a need to understand that exposure systems are dynamic when it comes to particles. It's not like chemicals where you typically would get like an even, they dissolve and you get this even dissolution and exposure for chemicals in exposure systems. For particles, that's typically not the case. They will float, they will sink, they will aggregate. You know, they will not be equally dispersed or spread throughout the exposure system. And then we have the issue of selecting the most appropriate species and life stages. I'll come to that a little bit more, but that's also an important point. And then the final thing is we need to understand what endpoints are relevant to study. It's not surprising the way things are, but most toxicity studies are conducted using methods and exposures that were designed for chemicals. And it might be that we're not using the right endpoints or studying the right endpoints when it comes to particles. So conducting exposures and toxicity testing of particles represents a number of key challenges compared to this conventional assessment of chemicals. And perhaps one of the first things to think about is the polymer type. So certain polymers, particularly your polyolefins, polyethylene, polypropylene, they're buoyant in natural waters, so they're going to float. And you have things like polyester and PVC and other polymers that are heavier or denser than seawater even, and they will sink to the bottom. So straight away, you've got, in an exposure system, you've got some particles that are likely to float, some that are likely to sink. And you need to choose an organism that, reflects where those particles are going to be. So there's no point choosing a benthic test species if all your particles are floating. There are mechanisms, we know this, we find polyethylene and polypropylene particles and materials on the seafloor. So there are mechanisms to get these particles to sink, things like biofouling, for smaller particles, aggregation, both homoaggregation with themselves, but more often heteroaggregation with other types of particles can bring them, cause them to sink. And of course, you have the biological angle on this. Any organism that ingests plastic will repackage that plastic in its fecal material, and that's usually dense and will sink. So this raises that question, in toxicity exposures, where are my particles? And this is a very good example of where we need to have much closer links between toxicologists and analysts and environmental chemists, because we need to know what the actual exposure concentration was. If your organism's swimming around in the middle and the particles are floating or sinking, using a traditional mass-based dosimetric, 5 milligrams of particles per liter of water, that's not the exposure. So straight away, your experimental design is flawed because you haven't characterized the exposure properly. Again, if you're not characterizing the exposure, you don't know if you're getting aggregation occurring with the particles. That will also change the exposure and how the organism will interact with the particles. So these are important things to consider, along with the selected organism being relevant for the exposure, as I mentioned before, pelagic versus benthic. A little bit on ingestibility. So at this point, I'm going to introduce Brian. This is Brian the fish. You'll be seeing a bit of Brian just for the next few slides. So there are different ways that Brian can end up with plastic particles inside him. One of them is passive ingestion. So he's swimming along looking for food like fish do, and he eats copepods. If those copepods have been eating small microplastic particles and they have them inside them, when he eats the copepod, he's also eating those particles and ingesting them indirectly or passively. Oops, sorry, wrong way. But you also have direct ingestion. So if you have particles that are, or plastic items at least, that are in the size range of the food for a specific organism, then at least certain organisms have been shown to, a lot of organisms are opportunistic. So if they see something that even remotely resembles food, they'll have a go at eating it. And that's why we find plastic particles and plastic litter inside the digestive systems of so many different organisms in the natural environment. And I think this is also quite important because I think this formation of biofilms, this microbial colonization of on the surface of plastic particles and plastic items, that changes how the particle looks to organisms. It's got if it has this biological coating, it can look more like food. And there's been studies that have shown increased uptake of particles that have got a biofilm on them compared to pristine particles. Then, of course, there's the size and the life stage. So this is Brian again, and he's fully grown here, and he's no longer eating copepods. He's eating smaller fish. So he's gone from potentially, he's now potentially taking larger plastic items, thinking that they're food. So, this is something that will change through the life stage, different life stages of an organism. And of course, size is important. This is Barbara. This is Brian's friend. She's a totally different species of fish, despite the fact she looks very similar. And she's much smaller when she's fully grown. So she still eats, or she eats zooplankton. Brian's eating fish. the prey size is totally different, but we're still assessing fish here. And then, of course, going back to life stage, Brian wasn't always a great big fish. He was a baby fish at one point, and when he was smaller, he would interact with particles of a much smaller size than when he's an adult. So the exposure to plastic pollution will change throughout an organism's lifetime. And we know that certain life stages, particularly the earlier life stages, organisms typically more sensitive to pollutants. I come back, I promised I would, to the plastic-associated chemicals. And we think in the community that this is really quite an important aspect and one that hasn't had enough focus to date. So if we have a plastic particle in the environment and that plastic contains some additives, here I've just used bisphenol A as an example because I think most people have heard of that. The chemicals can leach out of the plastic and into the surrounding water. And when that happens, if there's organisms present, then you would get what I would consider a conventional exposure scenario for the chemical. It's in the dissolve phase, the fish or any other organism is swimming through, and this exposure there and this potential for the chemical to be taken up and potentially accumulate, cause a toxic effect. But there is for particles, when particles are present, there's also a secondary exposure route, which is the direct ingestion of the particles containing the chemicals and the potential for these chemicals to leach out during transit through the gut of the organism, like this. So we still don't know how important this route is, but at least it is potential there. And we know that the gut conditions in many organisms are different to those in the natural environment. They're more acidic, or there's all sorts of differences in the conditions that might promote leaching during the transit through the gut. It's very important, though, I'm back on safer ground here, I'm a chemist. It's very important, though, to remember or be aware that just because there are chemicals in a particular material, it doesn't necessarily mean that that's what you're going to find in the leachate. I've just got an example here, car tires, try to mix it up a little bit. And when we look at car tires, we take a solvent extract, we can extract a lot of chemicals from the car tires. And then if we do a leaching study for 14 days in seawater, and then we analyze both of those samples, what we see is the following. So this is the solvent extract, and this is gas chromatography, mass spectrometry analysis. You see lots and lots of peaks, that sort of raised baseline. That's when you have so many chemicals present that we can't actually separate them. So huge numbers of chemicals present in the car tires, which you might expect to see in the leachate. But actually, the situation is quite different. It's only certain chemicals that leach out in high quantities, and these do tend to be the more polar and the smaller molecules, which kind of makes sense. Going back to this additive leaching in the gut of organisms, we did try to study this. So we took beach litter from a beach in the Netherlands, and we categorized it. We worked out what polymer type everything was. We looked at what percentage by mass, at least, it represented for the litter. And then we worked with the northern fulmar seabirds. These are very well studied. They're known to collect or ingest lots of large plastic items, but they also have a mechanism in their digestive system that grinds the particles down. So I think the estimate is something like between 30 to 90 days, they will grind a lot of stuff down into small particles that they can then excrete. So we know that they have potential for a lot of exposure to chemicals. So we cryomilled the beach litter and we incubated that with stomach oil from the seabirds for up to 90 days to mimic that length of time that they can be there. And Then we did the same approach. We took a solvent extract of the reference material, microplastic reference material that we'd made, so that we knew all of the chemicals that were present. So these were some of the ones that we were able to identify. And then from this, we developed a more specific analysis method. So we start with non-target screening, looking just to try and find what's there. And then we pick out some marker chemicals that we think we have good identification for, and then we develop a more dedicated analysis method for those. We apply that to the stomach oil to see which of these chemicals are coming out of the plastic and into the stomach oil. And I've just picked out one of the examples. It varied, not all of them were present in the stomach oil, but for certainly some of them we saw quite significant increases. So you've got the control sample in green, and then the blue line shows the concentration of this chemical TCEP in the stomach oil. So we were able to demonstrate that this, at least for certain species, this is a viable exposure route. So when we put all this together, the question is, for me as a chemist, I would always ask toxicologists, what is it that you actually just tested in your toxicity study? Because my experience from looking at the literature is that there's really limited physical chemical characterization of the test materials that are used in toxicity studies, particularly the chemical side. There's not been very much focus on the properties of the particles that are driving the observed toxicological impact. So I don't know whether it's a particulate effect, whether it's a chemical effect, and so on. And there's virtually no focus on looking into the mechanisms of toxicity. I'm just taking a look at time. I think I'll look to wrap up now. So I'll just give an example of this of a study that we just published this year. Excuse me. It's a collaboration that we had with a research group in China. And here we were trying to look at the differences of more spherical particles. They weren't as spherical as we'd have really liked, versus really regular shaped particles. So we wanted to see if there was a difference between these primary and secondary microplastic, and we used PVC particles. We purchased the spherical ones, and we created the irregular shaped secondary microplastic using milling. We isolated the same size range for both particles, so it's 53 to 106 micron, and then we used a couple of exposure concentrations, a high and low. We exposed these to Madaka fish embryos, and we studied some endpoints that we thought might be of interest. So this is O2 flux, some hypoxia responses, genes, cardiac development genes, and hatching genes. I won't go into detail, but I just want to say that the chemical contribution, we were aware of this, particularly with PVC. So it was validated and tested that the way we set the experiment up, there was no chemical toxicity here. It was a direct comparison on the physical toxicity. What we saw was a range of malformations for the different exposures. You can see the list here. So in the high secondary microplastic, that's where we saw the greatest malformations. And there we had spinal deformities and yolk sac edema and what was the other one? Pericardial edema were much higher in that exposure group than any of the others. When we looked at the primary versus the secondary particles, for the primary, we didn't find any particles inside the embryos. That's something we checked and we didn't expect to. So this must be a surface related issue. So what we saw with the primary, there was a high attachment to the chorion, the outside surface. There was no effect on hatching rate. There was a reduction in hatching time, and there was a slight increase in heart rate. The average oxygen influx was also significantly lower for the primary particles. When we looked at the same endpoints for the secondary, we saw that there was some attachment, but definitely less of the secondary particles. There was no effect on hatching rate. There was a much bigger reduction in hatching time, and there was a bigger heart rate increase. But there was the same or even a higher O2 influx into the chorion, into the embryo. When we look using SEM imaging at the surface of the embryos that are exposed to the secondary microplastic particles, we saw regions of damage. So the yellow arrows and the dotted area, that's damage that we could see on the surface, and then the yellow circles highlight the villus that were the chorionic villus that were damaged as well. So from this study, we're able to pull out some different physical toxicity mechanisms. So the first, for the primary microplastic, was reduced hatching time, slight heart rate increase, and malformation of the larvae. And then for the secondary microplastic, much greater reduction in hatching time, greater heart rate increase, and greater malformation of the larvae. And we were able to sort of hypothesize that it's reduced oxygen into the embryo due to this a higher coverage of particles for the primary, and then it's this physical damage from the irregular shaped particles on the surface of the chorion that is driving the differences in the toxicity. So I'll wrap up here. So what all this means is that with many of the micro nanoplastic toxicity studies employing these spherical particles that are either a single size or a very narrow size distribution, often just comprised of a single polymer type. That's what most of the toxicity data that we have available for microplastic is based on. And it becomes very, very difficult to extrapolate the results from those types of studies to this complex mixture of particles and associated chemicals that actually represents the microplastic that we find in the environment. So I'll stop there, I think, because it's time to stop.
Okay, thank you so much. Now we have some time for questions, so you may raise your hand or just unmute yourself. Yeah. Yes, Emily.
Hi, I'll turn my video on for a minute because I hate answering questions to people who are not looking at me. Just to make things even more complicated with the environmental representative studies, have you investigated the influence of weathering of the particles or biofilm formation on uptake and toxicity?
Yeah, so we kind of getting there in the community for two reasons. One, because we know that those are the types of particles that are more relevant. So we are starting to use cryomilling a lot more to generate microplastic reference materials, but it's hard to get particles below about 100 micron with cryomilling.
Yeah.
And for a lot of toxicity studies, you want to have done with invertebrate species that ingest quite small particles. So we're starting to actually combine UV degradation and cryomilling and different techniques to actually get smaller and smaller particles that we can use in toxicity tests in bulk quantities. And what that's starting to do is actually move us more towards an environmentally representative test material because it has this degradation.
Yeah.
There have been some studies with biofilm formation. I had a slide on it in here. It's not been studied extensively, but the available evidence suggests that it certainly promotes ingestion, but whether it affects toxicity specifically, it's probably unlikely. I mean, it's the microbes that these organisms are swimming through or living around all the time anyway, is the theory.
As a side note, I've worked with a group on microplastic out at Colorado School of Mines and Johns Hopkins, and they were doing cryomilling for plastics to create them, and we were able to get down less than 10 micron. It's just very difficult to detect. We were doing metal tech microplastic, so we were investigating them with ICP, but I know like your FTIR and your standard methods are really, really
Yeah, I mean, you
check
can,
those.
I mean, cryomilling absolutely produces some nanoscale particles, but
Yeah.
you know, we've done some pretty intensive cryomilling, and in terms of the mass that you start with, when you get down to sort of 1 micron and below, it's like 0.00001% of the start material is in. It's a huge number of particles, but by mass, it's a tiny amount relative to the start material. It's still, it seems to hang somewhere between sort of 80 to 100 micron is about as low as you can go for the bulk of the material.
Yeah, that makes sense.
Was that work then for using the metal dope particles? Was that Denise Mitrano's?
No, I actually worked with James Ranville, who was Denise Mitrano's advisor way back when. She is doing only full digestion of the particles. So she has the palladium dope particles and she's microwave digesting them, but we were doing single particle ICP-MS. We were getting the particle size distributions. So.
Yeah. I mean, that's it's such a challenge, you know. I mean, I think there's a real need for those doped particles for tracking and knowing that this is your particle and not something that, because the microplastic is everywhere, as we know. So contamination is always a challenge. So, but again,
but it's not good for environmental detection at all. Though I have noticed a lot of studies that are coming out that are looking at metals that were manufactured as additives into the plastics, and then they can use that as a track as well. Yeah.
So I don't work so much with metals, and I tend, because I'm an organic chemist by background, so I tend to forget the metals. But certainly when it comes to additives, there's a number of the additives are metal based, particularly some of the colorants and dyes that are used. So those, doesn't surprise me that those would make for good tracking, particularly with the sensitivity of ICP-MS instrumentation.
Great. Well, thank you for the talk. I appreciate it.
Very welcome. Thanks for listening.
Thank you. Now, Ganit has another question, and I just want to say to everyone that the webinar is being recorded and will be uploaded to the Chief Scientist YouTube channel. So, Ganit.
Hi, thank you. I would, according to the EU Drinking Water Directive, by 2024, the member states should adopt a methodology to measure microplastics in drinking water. Do you know how they're getting on with it?
I would guess that they're relatively close. Now, drinking water is a really easy one because it's a very simple matrix. And it also most likely will not involve particles that go right down to sort of 10 micron. There's probably likely to be a higher cutoff. I'm not 100% sure myself. I'm not involved in that work, but I know the guys who are leading a project called EuroQCharm, which is a coordination and support action funded by the EU, which is all about harmonization and standardization of microplastic analysis and things like that. So we've been sort of getting there. Whether it'll be quite ready for adoption by 24, but it won't be far off, I would suggest. I mean, we can do it now. It's more about the harmonization. We have the methodologies to be able to do it. It's just making sure that it's reproducible and harmonized so that you can go out and monitor and have comparable data. That's proving to be the hardest task.
Thank you.
Okay, so if there are no more questions, I think we can end this webinar. And thank you so much. You've been so nice, and it's been nice. The correspondence with you was very, so thank you very much.
The correspondence was really easy. It was good communication, so thanks for that.
Yes.
And it was my absolute pleasure to come and present. Thank you so much for the invitation. It's always an honor.
Yes.
Invited to such things. So, and it was even nice to see a familiar face in the audience, Issam.
Yeah, it was worth, to attend to see just only, your photo as 16 years, with hair.
So
Yes,
yeah,
it's
Issam and
also
I
been-
worked together for a few years. Does everyone know Issam?
I know the Gani here, I
Yes,
guess.
we know each other from the Technion.
From the
Hi.
Technion, yeah. And
Nice.
Matan,
If
yeah.
you remember, we met also in
Yes, Matan, yeah. I know a few, yeah. When
all the Corona, so everything forgotten.
Yeah, it's like a reset.
Mhm.
So yeah, we've been working, trying to publish some papers at the moment, aren't we? So,
Yeah.
from one of our projects.
What happened to the jellyfish project?
Yeah.
That's the one.
Yeah. I guess Matan we met in Italy, yeah.
Yes.
Yeah, that's it. Okay.
Cool. Well, it's nice to see everyone. Thank you again for the
Yeah.
invitation. I have to hop off the call because I have a meeting.
Yeah, we need to end it now. So thank you very much for
Bye bye.
the
Thank you.
audience. Bye bye.
Bye.